In the next 2 lectures, we'll discuss biological models and then experimental techniques used to deploy those biological models. We'll begin here with an introduction to those biological models. At the end of this lecture, you should be able to explain how biological models are used to complete an ecological analysis. And towards that end, you'll be able to define what is an ecological analysis. Your reading is chapter 23 from the textbook. There are several kinds of biological models available to us in rumen microbiology. We can use cultures, nautobiotic ruminants, defaunated ruminants, and in situ nylon bags. The goal of these biological models is to carry out an ecological analysis of the rumen. To define what is an ecological analysis, we'll refer to this text from Robert Hungate. He said, "The following questions arise in a complete ecological analysis of a natural habitat. What kinds and numbers of organisms are present? This involves identification, classification, and enumeration. What are their activities? Food and metabolic products must be identified, and habits of growth, reproduction, and death known. A complete determination of activities necessitates a complete knowledge of the environment. And to what extent are the activities performed? This involves quantitative measurement of the entire complex as well as individual components." In short, by carrying out an ecological analysis of the rumen, we're answering who's there and how many, what can they potentially do, and what are they in fact doing. To discuss biological models, we'll begin first with cultures. We'll organize our discussion of cultures by discussing different types and techniques. This categorization is somewhat arbitrary. Here, we'll define types of cultures as pure, defined mixed, and undefined mixed. We'll define techniques for cultures as either batch or continuous. After this class, I don't want you to quote different types and techniques of cultures as shown here. Again, their classification here is somewhat arbitrary and used just for organizational purposes. Pure cultures are the ones that have one species cultured in isolation. This is a method for identifying predominant bacteria and their niches. You're familiar with pure cultures because it's what we've been studying most of this class. This is what's covered in Table 9-1 in the Russell textbook. As we've discussed before, pure cultures are established first by inoculating media with rumen contents. We then serially dilute and then pick colonies at high dilutions, around 10 to the 8th, where we can start to see distinct colonies, and establish pure cultures from those colonies. We then study phenotypic characteristics and assign species and functions. Here we see a roll tube culture of Strep bovis. Each of the tubes represents an increasing 10-fold dilution. You can see at the tube at the very right, we're able to start to see distinct colonies. This is a pure culture already, and we are doing dilutions just to check for contamination. But if this were a sample of rumen contents, we would pick one of the distinct colonies, inoculate a culture, and begin the phenotypic characterization. Most of the pure cultures we study today were established decades ago in the 1950s and the 1960s by 2 scientists and their students, Marvin Bryant and Robert Hungate. The strength of pure cultures is that we can unequivocally assign characteristics to one species. Other strengths are that they're simple and they're reproducible. We can take the same pure cultures that were first established in the 1950s and 60s, and we can do the same experiments with them and get the same results as Marvin Bryant and Robert Hungate did decades ago. The weaknesses of pure cultures is that they're biased towards organisms that are at high abundance or thrive in high nutrient concentrations, by virtue of how we establish the cultures. We're picking colonies at dilutions that are 10 to the 8th or higher, where we can start to see distinct colonies. It's unlikely we'll ever pick a colony representing an organism in low abundance, one with a concentration of much less than 10 to the 8th per milliliter. We're also biased towards organisms that thrive in high nutrient concentrations, the same as the conditions that we use in our media. If we use media with lower concentrations, as we've recently done, we can get different organisms, but this is an avenue that we need to further explore. By studying a single species, we're also ignoring microbial interactions. Defined mixed cultures are combinations of more than one pure culture. For example, we can have two cultures, which are co-cultures, or we can have tricultures, made up of three cultures. We saw earlier a co-culture between Ruminococcus albus and a methanogen. The strengths of defined mixed cultures is that simple microbial interactions can be modeled, like interspecies hydrogen transfer between a bacterium and a methanogen. A weakness of these cultures is that the interactions are still simplified. Practically, it's hard to make a culture that has more than just a few organisms. Another weakness is the source organisms are those from pure cultures. They're the ones that are found in high abundance and thriving under high nutrient concentrations. Undefined mixed cultures are usually referred to as just mixed cultures. These contain multiple species, but their identity is unknown. To make an undefined mixed culture, we first inoculate media with rumen contents and then culture from that directly. We can process the rumen contents in several ways. We can dilute, we can use selective media to enrich for, say, glucose-utilizing organisms, we can wash, or we can fractionate cells. However, we cannot isolate pure cultures. If we do this, then we have a defined mixed culture by definition. The strengths of undefined pure cultures is that they include organisms that cannot be isolated in pure cultures. Because more organisms can be present, they can represent more complex interactions. The weaknesses is that they're hard to produce. Rumen inoculum changes over time, and it differs across animals. Another weakness is that it's hard to assess the activity of individual species, by virtue of having so many species and by having an undefined culture. We don't know everything that's present. Each of these types of cultures can be deployed using one of two techniques. We can use batch cultures, or we can use continuous cultures. Batch cultures are oftentimes called in vitros, though continuous cultures are also in vitro systems. In batch cultures, we perform an incubation in a closed system with a single batch of media. These are often used to determine fermentation end products and substrate degradability. For example, the degradability of a test feed. Their strengths are that they're simple. We use a single batch of medium, we harvest the cultures at some later time point, the experiment is done. The weaknesses are that they accommodate only short-term experiments, those lasting hours to days. The medium is not changed, and so the short-chain fatty acids accumulate. These can decrease pH or inhibit fermentation. We have difficulty in adding substrate repeatedly exactly because the end products accumulate. We normally give one large addition of substrate at the beginning, just large enough to last the microbes through the end of the experiment. A continuous culture is also called a chemostat. It involves incubation in an open system with continuous addition of substrate and removal of end products. It's used to determine fermentation end products and substrate degradability, just as batch cultures. However, another measurement particularly appropriate for continuous culture is microbial efficiency. We can measure microbial efficiency better with continuous than with batch culture experiments because, as we'll discuss, we can keep continuous culture experiments running for long times. The microbes are in a state of good health, they're not in a state of dying like in batch cultures. Continuous culture systems used for rumen microbiology have certain modifications. Most of these modified systems have the ability to add solid substrate, like a pelleted feed. For conventional continuous culture systems in microbiology, only liquid medium can be added. Another modification for rumen microbiology applications is the separate removal of liquid and solid from the culture system. This permits liquids to be removed faster than solids, mimicking the situation in the rumen. A conventional continuous culture system would not enable this separate removal, and liquids and solids would be removed at the same rate. The strengths of continuous cultures are that they can accommodate long-term experiments, ones that last days to weeks. Substrate is continuously added, end products are continuously removed, microbial growth can occur indefinitely. The weaknesses is that continuous cultures require a long adaptation to get to a steady state. normally a few days at least. With batch cultures, in contrast, you begin the experiment at the time of inoculation. Another disadvantage is that they're very laborious. It takes one person full-time to maintain a continuous culture system like we saw on the previous slide. They have a lot of moving parts, and they're liable to break at any time. A batch culture, by contrast, you simply inoculate and leave alone. Cultures are one type of biological models. Another useful biological models are nautobiotic ruminants. The term nautobiotic derives from the Greek nostos, known, and biotic, life. We know exactly which microbes are present in these ruminants. To prepare a nautobiotic ruminant, first we raise a calf germ-free. We deliver it by C-section and then place it in a sterile isolator. We saw this figure in the slide about microbial colonization. The rumen is then inoculated with pure cultures to establish a defined population, hence the terms known life. Alternatively, the calf can be briefly exposed to environment, or diluted rumen contents can be added. This creates an undefined but still a simple population. Technically, these animals are not nautobiotic, but meroxenic. These ruminants can be used to study many problems, including the development of populations. For example, it was found that Fibrobacter succinogenies colonizes only if more than 100 other strains were first added. These colonization experiments couldn't be performed with any other model. They've also been used to study feed degradation. We saw these data earlier when we discussed interspecies hydrogen transfer, and we saw that dry matter degradation is faster when methanogens are present. As you recall, animals with methanogens absent showed lower degradation of straw between 24 to 72 hours compared to animals in which methanogens had been added. Another insight of studying feed degradation with nautobiotic ruminants is that cellulose degradation by pure cultures of cellulitic bacteria is similar to conventional animals. Meroxenic sheep were colonized with Fibrobacter succinogenies, Runococcus flavifaciens, or Runococcus albus. Conventional animals were included for comparison. With these sheep, cellulose disappearance was measured after 48 hours for ryegrass, wheat, and ammonia-treated wheat straws. For the animals inoculated with one of the cellulitic species, cellulose disappearance rivaled or was even greater than that for conventional animals. For the ryegrass, Runococcus flavifaciens led to a cellulose disappearance pretty close to that for the conventional animals. For the wheat straw, all three cellulitic species led to a higher cellulose disappearance compared to the conventional animals, and for the ammonia-treated wheat straw, Fibrobacter succinogenies led to a higher cellulose disappearance. This experiment is critical because it supports the idea that the three cellulitic species that we've isolated in culture are good models for cellulose digestion. Certainly, there are more species than just these three that degrade cellulose in the rumen, but these cultured species are able to do it just as well as the total cultured and uncultured ones found in the conventional animals. A final application of nautobiotic ruminants is to study microbial interactions. These have been used to study interspecies hydrogen transfer involving acetogens. Nautobiotic animals with acetogens present were pulse-dosed with hydrogen. When methanogens were absent, acetogens accounted for 25% of the total utilization of hydrogen, showing the acetogens could use hydrogen. When methanogens were present, however, the acetogens did not use any of the hydrogen, showing that they were outcompeted by the methanogens. This dovetails with our discussion of acetogens in the lecture on manipulation of fermentation. It confirms that acetogens are outcompeted by methanogens and are not a viable way to reduce methane. The strength of using nautobiotic ruminants is that it combines the versatility of cultures with the natural environment of the rumen. There are some questions that can be answered only with these ruminants. They have to be maintained in isolators under aseptic conditions and fed sterile feed. It is technically difficult, and there's only one group left in the world that can still do this work. Next in our discussion are defaunated ruminants. We had referenced defawnated ruminants earlier. These are animals that have had their protozoa removed. Rumen contents are emptied, the rumen wall is washed with a detergent, and feed is withheld for 24 hours. After feed is reintroduced, the animal is isolated to prevent it from being recolonized with protozoa from others. By comparing the responses of these animals with conventional animals containing protozoa, we can infer the function of protozoa. The use of these animals led to the conclusions we saw earlier in our lecture on rumen protozoa. It's found that defaunation causes decreased fiber degradation, protein degradation, and starch degradation, as well as animal live weight gain when energy is limiting. Further, defaunation increases efficiency of microbial protein synthesis and animal live weight gain when protein is limiting. The strength of using defaunated ruminants is it's the only live animal model for inferring protozoal function. The treatment used to remove the protozoa is harsh and causes more changes than just the removal of the protozoa. The bacterial community that recolonizes a defaunated ruminant is different from a conventional animal. The final biological model we'll discuss are in situ nylon bags. With in situ nylon bags, feed is incubated in the rumen inside a bag like that shown to the right. It's used to measure degradability of feed. A key point is the selection of bag pore size. The bag pores should be large enough to allow microbes to pass into the bag and thus initiate fermentation of feed. However, the pores need to be small enough in order to retain undigested feed. The strength of this technique is that the feed is exposed to a reasonably representative community of microbes, the full community of the rumen. Another strength is that degradability measurements are reasonably quantitative. Because we're exposing the feed to a representative community of microbes and conditions that are representative of the rumen, the degradability measurements are reliable. The weaknesses of the bag is that they're hard to assess the activity of individual microbial groups. We're exposing the substrate to everything in the rumen. Another weakness is that optimal pore size is difficult to achieve. In practice, we make a compromise. A typical pore size is about 50 micrometers, which prevents the largest protozoa from passing in, but also prevents most of the small feed particles from passing out undigested. A final weakness of in situ nylon bags is that degradation of soluble nutrients cannot be studied. Soluble nutrients pass from the bag pores without being fermented. In discussing the myriad of biological models, let's not lose sight of our original goal, which is to carry out an ecological analysis of the rumen. We want to answer who's there and how many are there, what can they potentially do, and what are they in fact doing. The take-home message from this lecture is that no one biological model can suffice. Each model has its individual strengths and weaknesses, and each operates at a different level of complexity. The figure at right attempts to order the models with increasing complexity as we go from top to bottom. This ordering is a bit contrived, but it tries to drive home the point that the models are complementary, and only together can we use them to carry out an ecological analysis and understand how the rumen works. In the next two